بسم الله الرحمن الرحيم. السلام عليكم جميعا، كل عام وانتم بخير بمناسبة حلول شهر رمضان الكريم. Today we'll talk about the last topic in molecular biology course, which is the synthesis of proteins. We'll talk about translation in two lectures, 1 and 2. Again, you can use this lecture as a main source for your reading, and you can back to Cooper textbook chapter 9 for additional reading. At the beginning, let us start with general information and concepts about translation. Protein synthesis involves interaction between three types of RNA molecules. Actually, the tRNA or the transfer RNA, the ribosomal RNA, and the messenger RNA. The tRNA, which is a kind of RNA molecules that are responsible for decode the messenger RNA sequence into a protein. The ribosomal RNAs, they are part of ribosomes, the factories of protein synthesis. We'll know about ribosomes in more details in the coming slides. And you know the messenger RNA templates, which is the RNA sequence that is a code for the protein synthesis. Now in this figure you can see the components of translation complex. Here we have the ribosomes. The ribosome composed of small ribosomal subunit and large ribosomal subunit. The small ribosomal subunit responsible for binding of messenger RNA and the large ribosomal subunit. The large ribosomal subunit responsible for binding to the tRNA and the small ribosomal subunit. You can see here we have the messenger RNA and how it interact with the ribosomes. Here you can see three different sites. The exit sites here, the polypeptide sites, the P site here, the amino acid site, which is the A site. The A site is responsible for receiving or accepting the tRNA charged with amino acid. The P site responsible the site where the polypeptide synthesis occurs and the growing polypeptide started, like as you can see here. And we have the exit site. It is the site where the tRNA removed and again used for another amino acid again. So, there are some important concepts that you need to know. In transcription, the DNA sequence of a gene is rewritten in RNA sequence. So the sequence of DNA or a gene is rewritten or transcribed into RNA sequence. In translation, the sequence of nucleotides in the mRNA is translated into amino acid sequence or polypeptide, which is later on can form proteins. So as you can see here that the number of nucleotides in the gene is proportional to the number of amino acid in the protein. We call it a colinearity of genes, mRNA, and proteins. So when you have usually or in general, when you have a longer DNA sequence or a gene, then you will have longer mRNA sequence and you will have a longer polypeptide chain. So that's why it's called linear. Here in this figure, you can see that the mRNA is read by a tRNA in triplets. What we mean by triplets? Cells decodes the mRNA by reading their nucleotides in groups of 3. We call it codon. So for each amino acid we have a codon, and the codon containing or consists of 3 nucleotides from the mRNA. Okay. حكينا انه our cells decode mRNAs by reading their nucleotides in groups of 3. لما احكي decode بقصد انه المقصود فيها انه يترجم. اذا we have a codon for every amino acid, and each codon contains 3 nucleotides. So, if every codon contains 3 nucleotides, so how many possible codons that we can get? خلينا نحسبها ببساطة. Look at this table. You can see here, for example, to this code. Look at the first nucleotide. How many possibilities you expect? How many possible nucleotides to be there? We have here U, but it can be C or A or G. So we have 4 different possibilities, right? U, C, A or G. The second nucleotide also we have 4 different possibilities. So we have possibly U, C, A or G. And the third nucleotide also we have 4 different possibilities. So 4 by 4 by 4. So 4 to the power 3, which is 64 different codes. So we have 64 possible different codes that can be formed for protein synthesis. But not all of these codons specify amino acid. Most of them specify amino acids, as you can see here in this table. But some of them specify a stop codon, which is not for a specific amino acid. It is just a stop codon that tells the translation machine to stop and terminate the synthesis of the protein. Also, we have a start codon, which is the AUG, which encodes a methionine amino acid. It is a signal for the translation machine to start the synthesis of protein. So we have start codon. We have also stop codons that stop the protein synthesis. Also in this table, you remember that we have only 21 amino acids in our bodies, in eukaryotic cells. So for each amino acid, possibly we have, or we can have more than one codon. For example, for leucine amino acid, we have 6 codons, as you can see here, that can specify the leucine amino acid. Also, we have 4 codons that specify the valine amino acid. So it's possible that we have more than one codon for each amino acid. Remember this. بدناش اياك تحفظ هذا الجدول مرة ثانية. بدناش اياك تحفظوه بس تفهموا ال principle or the concept of code. And there are possibilities for more than one codon for each amino acid. And we have the starting codon, which is the AUG, specified methionine amino acid. We have stop codons, 3 stop codons. UAA, UAG, and UGA. They specify the termination or the stop of the translation. Now, let us talk about the general features of genetic code. Again, we have 64 possible different codons. You should know also that the translation or the reading of mRNA, it should be from the 5' to the 3'. So our translation machine read the mRNA from 5' to the 3'. You remember that in transcription also the synthesis of new DNA is from 5' to the 3'. Also in the transcription the synthesis of mRNA also from 5' to the 3'. Again, we have 61 codons specify an amino acid. We have 3 stop codons, UAA, UAG, and UGA. They do not encode any amino acid. They are just a signal to the translation machine to stop the protein synthesis or the translation. In the mitochondrial DNA, تذكروا mitochondrial DNA, which is a DNA exists in the mitochondria. We have the same, almost the same universal code. What do you mean by universal code? Universal code, that means this code or this table can be applied to almost all living organisms. So these codes is fixed or are fixed for all species تقريبا. خلينا نحكي عن تحدثنا عن protein translation general concepts. هلا خلينا نتذكروا حكينا عن mutations and how the mutations in the DNA or in the genes can be translated into the protein. For example, خلينا نحكي عن single point mutations. تذكر point mutation is the substitution of a single base, so changing a base from one to another. They are divided into different types. For example, the silent point mutation. The silent point mutation has no effect on the protein sequence. What do we mean by that? As you can see here, for example, we have a sequence or DNA sequence. We have the mRNA sequence transcribed from the DNA, and the amino acid sequence translated from the mRNA. Let us say, for example, if there is a mutation occurs here in A and substituted into T. The substitution of A into T here will change the mRNA sequence, as you can see here, and will be also reflected or راح نشوفه أيضا في الأمينو أسيد sequence الناتج. لكن لما يتغير أو يكون السبستيتيوشن أو البوينت ميوتيشن غيرت كود زي الجي يو اي للفالين غيرته لجي يو يو ايضا ظلوا فالين، لانه بتذكر هون في التيبل نذكر هون على الفالين we have 4 different codes، لكن هون التغير في هاي النيوكليوتايد ما ادى الى تغيير الامينو اسيد سيكونس، لانه تغير من كود لكود لنفس الامينو اسيد. So it changed from code that specify into another code that specify the same amino acids. That's why we call it silent. So this point mutation will not affect the final amino acid sequence. The second type of point mutations, we call it missense mutation. And in the missense mutation, the results in the amino acid substitution. So we have a change in the amino acid sequence. You can see here, for example, in this, the threonine, for example, and proline. In the normal DNA sequence, usually we should get from the CCC code a proline. But if the C at this position substituted into A adenine, like this one, you can see that the code ACC is different from the CCC. So the CCC specify the proline amino acid, while the ACC specify the threonine amino acid. That will change the amino acid sequence. So just changing a single amino acid in the polypeptide chain or the amino acid sequence. Now, here there is something important. Some of you may ask the question, if we have the missense mutation and we have the change in the amino acid sequence, in the substitution in one amino acid, so we have different sequence of amino acid, is that will induce a disease or will harm the function of the resulted protein? Actually, that depends. إذا كانت الميوتيشن أو substitution جاي في أمينو أسيد، هذا الأمينو أسيد بيلعب دور مهم جدا في الفانكشن للبروتين، أو غير في تركيبه بشكل مهم مما أثر على الفانكشن تبعته، أكيد راح يؤدي لتأثير على وظيفة البروتين. زي substrate binding site أو catalytic sites أو إلى آخره. إذا تغير الأمينو أسيد هناك في change, then it will harm the function of the protein. But if the amino acid change in the body of the protein in something not harming the function of the protein, then this substitution will not harm the function of the protein, and the protein will stay doing the normal job. إذا وعلى ذكر هاي النقطة أيضا بدي إياكم تفهموا نقطة مهمة جدا. في شيء اسمه دائما structural and functional relationship. What you mean by structural and functional relationship؟ بمعنى إنه the function أو الوظيفة تتناسب مع التركيب أو الشكل. على سبيل المثال for example the wheels of the car، عجلات السيارة، تخيلوا إنهم لو كانوا مربع مثلا مش دائرية، فإذا الشكل الدائري تناسب مع الوظيفة. there is a relationship between the structure and the function. If the structure changed, possibly the function will be affected. نفس الفكرة هون. إذا كان الأمينو أسيد وتغيره أثر على structure and maybe will affect the function of the protein. The third type of point mutation, we call it nonsense mutation. So a normal codon for a certain amino acid can be changed into a stop codon. So as you can see here, for example, here the substitution of of C into G. So the codon in the normal DNA, we have the code for the tyrosine, for example, UAC, and here we have it, if it's substituted into G, then it will change into UAG, which is a stop codon. That will result with incomplete polypeptide chain and immature protein. Then mostly this resulted amino acid sequence or later on protein will not do the proper job. So the substitution of normal codon into stop codon, as you can see here, we call it nonsense mutation. Also, we have the insertion or deletion mutation. What do you mean by that? If you delete or insert, إذا أزلت أو أضفت nucleotides، هاي بتؤدي إلى frame shift in the protein synthesis, or frame shift mutation. So let us say this example. For example, we have here the GTA, and we have kind of deletion for the T and A. So if we remove the T and A, that will end with a different nucleotide sequence, as you can see here. So the normal one should be serine, valine, proline, tyrosine, and the new one will be serine, alanine, leucine, and leucine. So as you can see, removing two nucleotides from the original or the normal DNA will cause a change in the code of amino acids and producing different amino acid sequence. And this different amino acid sequence possibly will be not normal and do no proper function. Now let us talk about the tRNA structure. So what is tRNA? tRNA are short, single-stranded RNA molecules with a size around 80 bases long. The charged or the activated tRNA can carry one amino acid, and this amino acid attached to the ribose of the terminal adenosine at the CCA at the 3' end of the tRNA. The attachment of amino acid to the tRNA usually mediated by a type of enzymes called amino acid tRNA synthetases. In fact, the specificity or the specific attachment of one amino acid to the tRNA is specified not only by the anticodon, but also we have some identifier sequences within the tRNA can specify the attachment of specific amino acid to the tRNA. Also, we have very important job in this regard for the amino acid tRNA synthetase. We have one amino acid tRNA synthetase enzyme for each tRNA, and it is proper amino acid. So the amino acid tRNA synthetase play a very important job in the linking of the amino acid with its proper and correct tRNA. Also, the tRNA contains stem loop structures, as you can see here, some modified bases, and some unusual bases like inosine. Inosine usually kind of unusual bases other than the normal ones, the C, G, A, and U. So the inosine produced usually from the hydrolytic deaminase or deamination through removing the imine group from the adenine and producing inosine, as you can see here in this figure. The stem loop structures, the modified bases and the unusual bases playing very important role in the stability of the tRNA, and also in the binding of tRNA to its proper codon in the mRNA. I don't think you need to know exactly what kind of modified bases and the positions and what they exactly do in each tRNA. I need you to know only that we have some modified bases and unusual bases. Also, we have the stem loop structure. We need to know the general structure of the tRNA only. You remember we talked about the codon and the anticodon, so we need to distinguish between them. In tRNA, we have three nucleotide sequence known as anticodon. That pairs with the codon or triplet in the mRNA molecule. You can notice here that the alignment is anti-parallel between the mRNA and the tRNA. You see that from 5 to 3 in the mRNA, so we have it complementary from 3' to the 5' in the tRNA. This is important for you to know. And let us ask the following question. What makes sure the accurate translation? Do we have mistakes in the translation? Actually, we have two steps required for the accurate translation. Adding the proper amino acid in its proper position in the polypeptide chain. So, the first step, which is the association of the amino acid to the tRNA by the amino acid tRNA synthetase. You remember that we have one amino acid tRNA synthetase for each amino acid and tRNA. And this amino acid tRNA synthetase is responsible for linking between the amino acid and it is correct tRNA. But sometimes the amino acid tRNA synthetase do mistakes, بيعمل أخطاء, and can bind to the wrong amino acid. لكن الامينو اسيد tRNA synthetase has its own proofreading site. So if there is a wrong amino acid, if the amino acid tRNA synthetase bind to the wrong amino acid, it will pops up the wrong amino acid back off and allow the correct amino acid to bind to it is correct tRNA. So the amino acid tRNA synthetase has its own proofreading capacity. The second step, which is the correct matching between the tRNA anticodon and the mRNA codon. تذكروا في عندنا التريبلتس في الكودون أو الانتيكودون لازم الانتيكودون should match and be complementary to the codon in the mRNA. لكن السؤال التالي is it a must that the anticodon should match completely the codon in the mRNA? Actually, no. We have some degree of flexibility in the third base of the codon. We call this wobble base pairing. Wobble base pairing. Actually, in wobble base pairing, we should have the first and the second bases, as you can see here in this figure. The first and the second bases in the anticodon should match and be complementary to the codon, the first and the second bases in the codon. While the third base, as you can see here, has some degree of flexibility. Let us understand more the wobble base pairing through the examples appear here in this slide. بتذكر حكينا انه for each amino acid sometimes we have more than one codon. For example, we have 6 codons for leucine amino acid, and we have 4, for example, for the proline and 4 for the serine amino acid. Now, how the wobble base pairing applied to these different codons for the same amino acid? Let us take leucine as an example. So we have identical leucine tRNA with the anticodon GAG. Normally, the anticodon in the tRNA or the leucine tRNA should pair with the codon CUC. As you can see here, this is a codon for leucine. But sometimes the same tRNA can pair with the CUU codon through wobble pairing or wobble base pairing. Actually, this can occur for the third base of the codons for the same amino acid. What kind of benefit can such wobble base pairing can provide? تذكر حكينا عن اللي هو silent mutations. Sometimes if there is a mutation occurs in the third base of the codon, that will change the nucleic acid sequence, but will not change the codons for the amino acid sequence. So just changing, for example, the U into C will provide the same amino acids. So the wobble base pairing can work like a buffer against deletion mutations. Also, the wobble base pairing again provide kind of flexibility for the synthesis of polypeptide chains. Now, let us talk about the ribosomes. Ribosomes are the factories of protein synthesis, the sites for the protein synthesis. In Escherichia coli, we have around 20,000 ribosomes, and they account around 25% of the dry weight of the cell. It is large quantity actually, as you can see. In mammalian cells or rapidly dividing mammalian cells, we have around 10 million ribosomes for each cell. So as you can see, we have large quantities of ribosomes in prokaryotes and eukaryotic cells, because we need the synthesis of proteins and these ribosomes involved in the all proteins synthesis. Here you see the figure again for the ribosomes. We have the large ribosomal subunit and the small ribosomal subunit, the mRNA and the growing polypeptide, as you can see here. Actually, there is an enzymatic activity in the large ribosomal subunit. We call it peptidyl transferase enzymatic activity. And this enzymatic activity is responsible for establishing the bond between the growing amino acid sequence and the coming amino acid. So it forms the peptide bond between the growing amino acid sequence and the coming amino acid or the new amino acid came with a tRNA. Actually, also we have, you remember that we have ribosomal RNA in the ribosomes. The ribosomal RNA in the large ribosomal subunit playing very important role in the catalyzing of peptidyl transferase activity. Now, let us go deep into the ribosome structure and the difference between prokaryotes and eukaryotes. In prokaryotic cells, we have the 70S ribosome, and in eukaryotic cells, we have the 80S ribosome. Before talking about the components of the ribosomes, we should know the meaning of S unit. What we mean by the S unit. S unit, it is the Svedberg unit, which is a unit for the sedimentation coefficient. بمعنى آخر, or in other words, it measures the sedimentation rate of a particle. أو بكلمات أخرى تحسب سرعة ترسب الجزيئات أو المركبات. So, we expect that the larger particles will sediment first and the smaller one will sediment later. بمعنى إنه الجزيئات الأكبر راح تترسب أسرع من الجزيئات الأصغر. So, based on this, you can see that in eukaryotic cells we have the 80 and in prokaryotic cells we have the 70. So we expect that the eukaryotic ribosome is larger, a little bit larger than the prokaryotic ribosomes. The sedimentation actually depends on the size, density, and the shape of the particle. بيعتمد على الحجم والكثافة وشكل البارتيكل أو المولكيول. So, in prokaryotic cells, we have the 70S, and it's divided into two subunits, the large ribosomal subunit, the 50S, and the small ribosomal subunit, which is the 30S. The large ribosomal subunit, 50S, contain 23S and 5S ribosomal RNA and 34 ribosomal proteins. The small ribosomal subunit contain 16S ribosomal RNA and 21 ribosomal proteins. While in eukaryotic cells, we have the large ribosomal subunit and the small ribosomal subunit. The large ribosomal subunit, 60S, contain 28, 5.8, and 5S ribosomal RNA and around 45 ribosomal proteins, while the small ribosomal subunit, the 40S, contain 18S ribosomal RNA and around 30 proteins. Now, let us talk about the RNA synthesis and the processing of ribosomal RNA inside our cells. So, you remember that we have the 18S, 5.8S, and 28S ribosomal RNA, and we have the 5S ribosomal RNA. For the 18S, 5.8, and 28S ribosomal RNAs, they are encoded by a single gene with a length of 13 kilobase long. 13 kilobase long. We have some intergenic DNA like spacers in the transcription unit or the ribosomal DNA transcription unit. For the 5.8 and 28S, we have the RNA polymerase 1 responsible for producing the primary transcript, So the RNA polymerase 1 is responsible for the transcription of these three ribosomal RNA molecules. So, the RNA polymerase will first produce primary transcript composed of 40S ribosomal RNA. Then this 45S ribosomal RNA undergoes post-translational cleavage to produce at the end the 18S, 28S, and 5.8 ribosomal RNA molecules. So, the 18S ribosomal RNA associate with the small ribosomal subunit. The 5.8 and 28S ribosomal RNA will associate with the large ribosomal subunit. Also, you remember that we have the 5S ribosomal RNA subunit, which is associated with the large ribosomal subunit. And actually, the 5S ribosomal RNA encoded by different genes transcribed by the RNA polymerase 3. As you can see here, that the synthesis and processing of ribosomal RNA in our cells. Now, let us describe the general function and structural components of ribosomes. Generally, ribosomes facilitate the specific coupling of tRNA anticodons with the mRNA codons in protein synthesis. The RNA components are responsible for the catalytic function of the ribosome, and the protein components responsible for enhancing the function of the ribosomal RNA molecules. Now, let us describe the chambers of the ribosomal subunits and the function of each chamber in the ribosomal subunit. You remember we talked about the mRNA binding to the small ribosomal subunit. So we have a mRNA binding site for in the small ribosomal subunit. Also, we have the E, P, and A sites. A, which is the site for the amino acid tRNA binding. P, which is a site for polypeptide tRNA binding. And the E, it is the site for the exit of the tRNA. And here also we have a channel for the exit of the polypeptide or the synthesized polypeptide. So we have first the charged tRNA or the amino acid tRNA will bind to the A site, and in the B site we have the polypeptide chain that will link the new amino acid here, and the polypeptide chain continues to grow. The tRNA, after linking the amino acid to the polypeptide chain, will transfer into the exit site and will exit the ribosomal RNA and do charged again with the amino acid and again back to the A site for continuing the protein synthesis. So here the general structure and functions of the chambers in the ribosomal proteins. Now, خلينا نحكي عن the general mechanism of translation. The mechanism of translation contains three stages: the initiation, elongation, and termination stage. The direction of reading the messenger RNA is from 5' to the 3'. So the ribosomes read the messenger RNA from 5' to the 3', while the protein synthesis begins at the amino terminus and extends to the carboxyl terminus. You remember that in the amino acid sequence or the polypeptide sequence or the protein, we have the N or the amino terminus, which is N terminus, and the carboxyl terminus, which is the C terminus. Also, you can see here the ribosome's active site where the peptide bond form between the growing polypeptide chain and the coming amino acid. You should know that in both prokaryotes and eukaryotes, the translation starts at a specific initiation site, which is the AUG, a codon for the methionine. And it does not start at the first codon in the mRNA. So you should know that it is not just once the ribosomes find the messenger RNA, it will start immediately the translation. There is a start codon that specify the start of the translation, and this start codon is a codon for methionine. Usually we call it the initiator tRNA. Of course, we need energy during translation. We'll talk about the energy consumption during the translation process. بدي اياك تتذكروا حكينا في محاضرة transcription. We have untranslated regions in the messenger RNA. We have the 5' untranslated region and the 3' untranslated region. The 5' untranslated region found upstream of the initiation site in both prokaryotes and eukaryotes and contain non-coding RNA. Also, we have the 3' untranslated region, which is follow the any of the 3 stop codons. So these untranslated regions are very important in the regulation of translation. We will talk about these untranslation region and the function of the translation regions in the coming slides. First, I want you to remember that in bacteria, the mRNA is polycistronic. Again, polycistronic, what we mean? We mean that producing several or different proteins from a single transcription unit, from single mRNA. While in eukaryotic cells, we have only single protein produced from a single transcriptional unit. So remember to differentiate between the polycistronic in the bacteria or in the prokaryotes and the monocistronic expression in the eukaryotic cells. Now, let us describe how the ribosomes can identify and bind the mRNA. Actually, the ribosomes cannot bind directly to the initiation site in the messenger RNA. Actually, they bind to the 5' untranslated region and then scan for the initiation site and start the translation. And we have a difference between prokaryotes and eukaryotes. Starting with the prokaryotes. In prokaryotes, we have a sequence called Shine-Dalgarno sequence. This sequence found around 8 bases upstream of the initiation site. This sequence identified by a complementary sequence in the 16S ribosomal RNA, which is a component of the small ribosomal subunit in the prokaryotes. Here you should know very important information that for each protein produced from the polycistronic mRNA in the prokaryotes, we have a start site or initiation site. And definitely we have also a stop site or stop codon for each protein. So even if we have a single mRNA, but for each protein we have a start codon and stop codon that start and end the synthesis or the translation of each protein. But in eukaryotic cells, the small ribosomal subunit bind to the mRNA through identifying the cap. You remember that the cap in the eukaryotic mRNA. So the ribosomes or the small ribosomal subunit will identify the 7-methyl-guanosine cap at the 5' terminus. So the 5' cap of the mRNA in eukaryotic cells is very important for recognition by the small ribosomal subunits. This is the difference between the prokaryotes and eukaryotes by the means of small ribosomal subunit identification. And you can see clearly that the small ribosomal subunits identify the mRNA at the 5' untranslated region and do not bind directly to the initiation site in the mRNA. في السلايد السابق حكينا عن ال 5' cap and its importance for the recognition by small ribosomal subunit, so allows the translation initiation. But in some mRNAs there is another way of recognition by the ribosomal or by the small ribosomal subunit, which is through the internal ribosomal entry site, IRES. IRES is an RNA element found in the 5' untranslated region that can be directly recognized by the small ribosomal subunit or through first binding with eukaryotic initiation factor 4G, followed by recruitment of small ribosomal subunit. In fact, this is a 5' cap independent way of recognition. And this way of recognition usually found in viruses, in the RNA viruses, used by the RNA viruses. In many of the RNA viruses, we don't have the 5' cap, and they use the IRES to recruit the small ribosomal subunits and initiate the translation. But also we have some eukaryotic genes and and eukaryotic messenger RNA use this IRES for recruiting the small ribosomal subunits and initiate the translation. You remember that we talked about the importance of capping at the 5' end and the polyadenylation at the 3' end, and their rules and critical rules in the protein translation. You can see here that at the 5' cap there is a protein eukaryotic initiation factor for E that bind to the 5' cap, and this protein bind to eukaryotic initiation factor G, which is already bind to a protein called poly-A binding protein bound to the poly-A tail. So kind of bridging between the 5' and the 3' ends of the messenger RNA. And this complex is very important for the translation initiation in the eukaryotic cells. Now, what is the first amino acid? We talked about the first amino acid in the translation, which is always the methionine, and methionine encoded by the AUG, you remember. So this is in both eukaryotic cells and prokaryotic cells, but in most of the bacteria we have the informal methionine, which is methionine with a formaldehyde group at the N-terminus of the amino acid. So in both prokaryotes and eukaryotes we have the methionine, but in most of the bacteria we have modified methionine with a formaldehyde group at the N-terminus of the amino acid, as you can see here. So, building of polypeptide in the prokaryotes or eukaryotes should be performed through 3 stages: the initiation, the elongation, then the termination. Actually, all of these stages require protein factors that help and aid the translation process. In the next lecture, we will continue talking about the translation process and the stages of translation. وراح نحكي أيضا عن regulation of translation and some examples about how the signals transferred from the outside to the inside of the cell, and how such signals induce specific protein synthesis in response